All right, everyone, I'm gonna go ahead and get started now that it's time. My name is Marine Andriot. Hello, bonjour. I'm a master's student in the Barreto Lab at Oregon State University, and today I'm going to be talking to you guys about the abiotic stress-induced alternative splicing landscape in the intertidal copepod Tigriopus californicus. So many intertidal organisms have evolved robust physiological responses to fluctuating abiotic conditions such as pH, temperature, salinity, etc. But a fantastic example of abiotic resistance is Tigriopus californicus, which is a very well-studied intertidal copepod. Here in the forefront you have a blown-up image of a gravid female, basically one with an egg clutch. And then on the extremities, you can get an appreciation for the natural setting in which you can find these types of copepods. And I hope you notice that these are much more desolate pools than the previous ones on the slides, and for very good reason. So first things first, Tigriopus californicus can be found on the Pacific coast of North America, everywhere from central Baja California in Mexico to northern Alaska in the US. And they're found in these splash pools in the superlittoral zone, and that's above the high tide line. So what that means is effectively there's very little water replenishment in these pools, and that makes it so that the conditions in the pool are highly dependent upon the daily swings of photosynthetic activity and temperature. And so before we talk a little bit more about the various stressors that we're going to focus on, temperature and dissolved oxygen being the two main ones today, we have to unpack a little bit of terminology about dissolved oxygen, or DO. So today, when I refer to something as anoxia or anoxic, that's going to be the adequate amount of oxygen for physiology, and hypoxia being the inadequate amount of oxygen, and anoxia being the complete lack of oxygen altogether. So with that, we can start looking at some actual data. So this was collected from a field site in Strawberry Hill in Oregon with a mini dot logger, and this is showing DO and temperature fluctuations following a DL cycle over 48 hours here. And so the top curve is actually representing temperature, and the bottom curve is representing DO. And so what I want you guys to notice here is that as we go through the yellow portions of the screen, that's representing daylight hours, and that comes with high temperatures and normoxic conditions. And as we move into those white portions, which are nighttime hours, we can see that we have a dip in temperatures, as well as our normoxic conditions sliding into hypoxia, and finally into anoxia for several hours before daybreak. So you can imagine inhabiting one of these really stressful ecological niches will probably select for abiotic stress resilience. And so to kind of add to the pot, our lab found that copepods are missing important parts of the HIF pathway, or the hypoxia-inducible factor pathway. This is highly conserved in metazoans, and it basically is the master regulator and master sensor of oxygen, and also controls a lot of other abiotic stress responses. And so that kind of begs the question, if not HIF, then what regulatory elements has this system evolved to tolerate abiotic stressors? So to delve into this, let's look at what we do know. So that's how gene expression is affected by heat stress. What we don't know is how hypoxia affects gene expression, and we know even less about how heat or hypoxia affect other levels of gene regulation. So one other level of regulation that I'm very interested in is post-transcriptional regulation, and one of the processes that I would like to focus on today is alternative splicing, or AS. So alternative splicing is a mechanism where pre-mRNA introns and exons are effectively are selectively included or excluded from pre-mRNA, from RNA transcript isoforms, and that effectively creates multiple different ones per single gene, and that in turn creates different proteins, which allows for functional adaptability and phenotypic diversity. And so with this, I hypothesized that alternative splicing may play an elevated mechanistic role in the evolution of abiotic stress tolerance in Tigriobus californicus. Now, there are a lot of ways that transcripts can be modified and regulated, but I want to introduce mechanism number 1, so differential transcript expression, which I will be referring to as DTE. This is the idea that if we have a gene and the gene is the sum of its transcripts that are being expressed, under 2 different conditions, if a transcript has a substantial change in expression, this is what it might look like. So this would be differential transcript expression, same thing as differential gene expression, but at the transcript level. This can be true for multiple transcripts. And the thing that I would like everyone to notice is that this is an example of whole transcript expression, or whole gene expression, sorry. And so in this case, when the whole gene is expressed, these 2 isoforms are going to increase in expression, but their proportions are going to remain the same. However, you can see that overall gene expression has changed, and so this would be detectable on a gene expression level. As opposed to mechanism number 2, this is differential transcript usage, where here we actually have multiple transcripts being basically switching in expression, flip-flopping, where the proportions of the transcripts are changing within one gene. However, overall, this may not be detectable on a gene expression level. And so hopefully you guys can see why we need transcript level resolution for this type of analysis comparing DTU and DTE. And so if we want to ask the question, how do abiotic stressors affect gene regulation at the transcript level, then we need to characterize the transcriptome. So that is what we did. We took our San Diego copepods and we took broods as replicates. We had 3 conditions: anoxia, heat, and control, with 4 replicates each. Anoxia being a 20-hour treatment, heat being 35, and control being normoxia in 20 degrees Celsius. And the most quality replicate for each of these treatments was then prepared on a long-read in-depth RNA-seq PacBio platform. And that was analyzed using isoseq and TAMA. And this gave us a merged transcriptome of full-length transcript isoforms. And this is what we got. So out of the 16,000-ish genes in the copepod genome that we know of, 12,737 genes were detected with 28,424 isoforms. And of those things that were detected, these are the 2 chunks that are appearing on the screen. 40% only produced a single transcript, and about 42% produced multiple transcripts per gene. And of those, 35% were multi-exonic, and 7% were monoexonic. This is significant because of the multi-exonic genes within copepods, 35% is actually very elevated for the level of complexity in this organism. And it's actually on par with other related crustaceans such as the Pacific white shrimp, which is actually kind of cool. Okay, so thus far we had presence-absence of transcripts, but in order to quantify them, we had to do a second RNA-seq experiment, which involved quantifying short reads. And for this, I had 4 experimental groups: hypoxia, anoxia, control, and heat. Heat being isolated at 32 degrees Celsius. And then hypoxia being isolated at 0.5 milligrams per liter of dissolved oxygen, anoxia after 4 hours post-plateau at 0, and control kind of lining up at that total 5-hour mark for the total treatment of all 3 of these groups here. And then all 24 samples here were sequenced on a 100 base pair Illumina platform, and that was mapped back to the merged transcriptome from earlier using Callisto. And the transcript abundances from Callisto were then funneled into DRIMSeq for DTU analysis and EDGE-R for DTE analysis. It's very early days for these results, but this is what we have so far. So right now we're looking at the differential transcript expression, or DTE again, between treatments, and I want you guys to focus on this red bar right here. This is the amount of genes where one transcript is, where at least one transcript has a differentially expressed, where at least one gene has a differentially expressed transcript. And so what's notable here is that heat appears to rely heavily on DTE, more so than anoxia in blue and hypoxia in green. And also very cool is that many of the DTE genes overlap across treatment groups. Hold that thought. We're looking at the same thing now, but for DTU. And immediately apparent is that anoxia relies more heavily on DTU than heat. And there is also little overlap in the DTU genes between treatment groups. So I conclude that DTE must be generally used for response to stress, while DTU may be more stressor specific. And this really makes sense based off of how they're controlled. Looking at this a little bit differently, here we have the amount of genes that are using DTU in yellow, DTE in blue, and both of them in orange. And what I want to highlight here is is how few genes are actually using both mechanisms. And so this kind of begs the question, are the different sets of genes in a singular response serving the same functions? And so we put that to the test with GO-term enrichment analysis, comparing DTE and DTU genes. So these significant DTE and DTU genes are very functionally enriched for all stressors. And it's very early for this analysis, but so far we can tell qualitatively that there's little overlap in enriched GO terms between DTE and DTU genes. So far they appear to be serving different functions. Here, lastly, is just an example. There's a little bit of a mess up here, but let me explain. Of a gene that was that was using DTU in heat response. So this gene features 10 transcript isoforms. Here are their proportions, and this green and blue are supposed to be significant. There's a little bit more going on there than I'd hoped. But this is a zoomed-in version that just highlights the significant ones that flip-flopped, and you can kind of see that better. Those are those two. And this is a 15 kb gene, so that's a pretty big structural difference. So in conclusion, the copepod transcriptome has high alternative splicing. Heat seems to rely heavily on DTE, and anoxia seems to rely heavily on DTU. DTE appears to be more general, while DTU is a more stressor-specific response, and distinct sets of genes appear to be serving different functions. I would like to acknowledge the whole Barreto team, as well as Oregon State University and NSF, and I will now take questions. Yes, actually, yes. Like just briefly looking at it, what I have looked at, it tends to be the case that the shorter transcripts are used. That's a good question. Yeah.
Following up on that, did you happen to check to see whether they are functional isoforms?
We have not done any validation studies yet. This is like the first of its kind already. Yeah, but that would be upcoming. Yes.
Was there any interesting GO terms that you came across? Did you say what they're different between?
Yeah, so it was kind of what it looks like is that there's more general overarching stress response terms that came up for the DTE genes. And the DTU genes seem much more specific, like regulating particular metabolites or molecules, like much more specific. That's kind of the general difference so far that I made out. That's a great question. It looks like it's about time for the next person, so I'm going to sit down, but Thank you so much.
Hi, everyone. Thank you so much for coming. My name is Daniela Zarate. I'm a postdoc with Dr. Jessica Purcell at UC Riverside, and I'm excited to be sharing my research with you today on gene expression in a supergene system that mediates alternative colony social organization in an ant. So, to begin with, let's see if I can move my slides. Wow, the entire PowerPoint is in French. I don't understand. How do we move slides? Okay. Perfect. Thank you. Okay, so to kick it off, this is a story of gene expression, but it is a story of gene expression set within a supergene context. Supergenes, in brief, are these massive co-adapted genetic complexes that have been implicated in the origin and maintenance of complex traits, and they have been found in a number of various systems. Let's see. Oh my gosh, I'm having oh, there we go. They have been found in a number of systems and underlie a variety of traits from morphological to reproductive, ecological, even social traits. And supergenes provide one system or one neat answer to the puzzle of how complex traits are maintained, because they ensure the co-segregation of adaptive alleles, usually as a result because these are regions of suppressed recombination as a result of them potentially being formed by structural rearrangements or inversions within the genome, so just a reverse orientation in the genetic material. And in the ant social supergene ant world and system that I study, there are two alternative supergene forms, the M form versus the P form, with the M being exclusively found in these monogyne or single queen colonies, and the P form being found in at least with one copy in the individuals in a colony that has multi-queens or multiple queens per colony. And this social supergene has been found to be ancient and widespread across the Formica phylogeny. This is a small snapshot of the massive Formica phylogeny with about 90 or so species on it, representing over 20 to 30 million years of independent evolution. And this supergene has been co-localized to occur on chromosome 3, which has been shown to be the result of at least 3 massive inversions. So here I'm showing you in Formica salesi, which is our model system, one Formica species. I'm aligning, this is an alignment of the M form of the supergene versus the P form of the supergene, and you can see from the crisscrossing lines that that these represent 3 massive inversions at least. And we were interested in studying gene expression in one of the Formica ants from this phylogeny outlined in red here, Formica neoclara. And the reason why we're interested in this species in particular of the entire Formica phylogeny is because it has a very interesting supergene genotype combination that is a departure from the classic Formica salesi model that was the first system studied. So in Formica neoclara, just like in salesi, our classic model, the monogyne individuals are homozygous or have 2 copies of the M. But in the polygyne system, we have all 3 genotype combinations represented. Again, this is a departure from our classic salesi model in which the salesi does not have the MM homozygote within its polygyne colony. So this immediately, it's apparent that offers a great opportunity to compare gene expression across genotypes while controlling for social form, because we can compare all 3 of these at once in the polygynous context. So I was interested in testing some hypotheses. The first was the very elementary starting point of, do these genotypes differ in expression at all? And here I have just a representation of our hypotheses. So given a given gene with some exons, the null hypothesis being no difference across genotypes, with hypothesis 1 being some form of genetic differentiation or gene expression differentiation. And then we have all the way to the other end of the spectrum, the extreme of potentially having 0 gene expression in any given haplotype, here represented by this complete silencing in the P homozygote. And the reason why we might suspect that there would be complete silencing in the P homozygote is because, again, the P supergene was formed by inversions. Inversions have been shown to potentially lead to degeneration as a result of the breaking up of genetically coadapted loci at the inversion breakpoints that has been seen in other systems. So we were interested in to see if there was some silencing going on. We were also, of course, interested in alternative splicing, as Marine nicely, beautifully explained in the previous talk. Genes can be alternatively spliced to form different isoforms, and so we were potentially interested in the role of that in mediating the supergene differences. And again, we have the null hypothesis of no differentiation between genotypes, and then a hypothesis that there is differential isoform expression between genotypes. And of course, these two mechanisms are not mutually exclusive. So for methods, what we did was we sampled a number of workers from 8 polygynous colonies, extracted RNA from the heads, DNAs from the gaster, and the DNA was used to assign supergene genotype. And once the supergene genotype was known for these workers, a number of heads were sent off for RNA-seq from each of the representative genotypes. And I do want to note here that we did pretty much have a full genotype representation per colony. So each of the 3 genotypes were sampled from each individual 8 of the 8 colonies. So we were able to control for that as well. We did a classic bioinformatic pipeline of RNA-seq, de novo transcriptome generation, and using classic tools for differential gene expression. And I just quickly want to say that I have a lot of thanks for this RNA-seq tutorial from the Canadian Bioinformatics Workshop Group, which helped me a lot in this. So first results are, this is a heat map of differential gene expression. I'm showing you 400 of the most differentially expressed genes, or DEGs, I'll call them from now on. We have on the x-axis individuals and on the y-axis transcripts. And the first thing to notice is that the individuals tend to clump together, they cluster together, so there is similar gene expression profile across supergene genotypes. And the second thing to notice is that the homozygote M individuals tend to have higher expression. As you can tell, there's more yellow in these transcripts, and yellow corresponds to higher expression, whereas the P homozygotes have lower gene expression. Again, they're more composed of the purple, and then the heterozygotes are also pretty similar, but these tend to be either more black or yellow in terms of their expression and relation. And I also want to say that of these 400 differentially expressed genes, about half of them do map to chromosome 3, and the other half map to other chromosomes in the genome. So that was really interesting, given that chromosome 3 is just one chromosome. About half of these are mapping to that section. So in a pairwise comparison, this is a volcano plot, pairwise comparison of the homozygotes. We see here we have log fold change on the x-axis and false discovery rate or significance on the y-axis. And the first thing to notice is that there's a dramatic spread of transcripts. Each dot represents a transcript, significantly differentially expressed transcript. The colored dots are the ones that are significantly differentially expressed. The black dots are those that are not differentially expressed. And the first thing to notice here is this dramatic splash of transcript on the MM side. So these that are colored blue are the ones that are more expressed in the MM individuals. And likewise, the ones that are colored red are most expressed in the PP homozygote individuals. And there's, as you can tell, there's a lot that seem to be just more dramatically expressed in the MM individuals. And the ones that are higher up in the graph are the ones with the highest significance. Although we do have some transcripts that are extremely more expressed in the PP homozygote, so we do have those as well. And I also quickly want to say that it might look that there are more DEGs on the MM side, but if you actually count these, there's more DEGs on the for the PP homozygote. They're just probably more clustered close to here with significance that is it overlaps more with others. So you see physically more of these, but the numbers are tell a different story. So zooming out to the other two pairwise comparisons with the heterozygotes. So I just showed you this one here on the left, and the two on the right are the heterozygote pairwise comparisons. And it's enough to say that this middle one, the heterozygote versus the homozygote M, the volcano plot is relatively symmetrical, whereas the homozygote P and the heterozygote has more of a dramatic splash, volcano plot splash here, saying representing that MP individuals have transcripts that are more highly expressed in those individuals. And interestingly enough, I quantified the number of DEGs that are either completely silenced, meaning there's zero expression in these transcripts, or close to completely silenced. And about 6% of these DEGs are completely silenced in the MM individuals, whereas 21% of these DEGs are almost completely silenced in the PP individuals. And so these 6% silenced are usually the ones that correspond to the ones that are furthest on the outskirts of the volcano plots. Comparing the heterozygote and the MM homozygote, we have 0, so none of them are silenced relative to each other. And when you compare the heterozygote and the PP homozygote, again, we see a dramatic number of DEGs that are completely silenced in the PP homozygote. So initial evidence potentially that there is some kind of lowered expression happening in the individuals with the P inverted supergene. And analysis of shared overlap between unique and shared differentially expressed genes and isoforms. This is a Venn diagram where the blue oval has DEGs that are more highly expressed in MM homozygotes. The teal oval has isoforms that are more highly expressed in MM individuals. And the blue intermediate section are those that are shared between those two individual analyses. The same is reflected on the other side, but in terms of the PP biased genes and isoforms. So these are isoforms that are more highly expressed in PP and so forth. So the main takeaway here is that there are a high number of unique as well as shared DEGs and isoforms in this analysis. But what I find the most interesting is this circle here, which there's 98 transcripts in which the isoforms, some of them with the same gene parent, some of them are more highly expressed in MM and others are more highly expressed in PP. So this is initial evidence that there is some alternative splicing in contrasting patterns between the two supergene genotypes. So in sum, there is differential gene expression between supergene genotypes. M-carrying individuals seem to have greater levels of expression, although this is in terms of magnitude and not directly in terms of count. The individuals with the P haplotype potentially have lower expression as a result of degeneration, maybe because of the inversion. There's similar levels of unique versus shared genes and isoform parents, and there's initial evidence of alternative splicing and contrasting gene and isoform patterns. And I didn't get a chance to talk about the isoforms, but I would love to talk about that later. Feature directions, gene identity analysis, continuing to map where these DEGs fall in the genome or the supergene, and I'm interested in also doing a allele-specific expression in the heterozygotes, and seeing if there's dosage compensation happening in the heterozygotes. And so with that, I don't think I have time for questions, but thank you so much.
Hello everyone, thanks for coming. Today I'll talk about gene expression in a hybridization perspective. And I apologize in advance if you have seen me present any of these similar ideas before. I'm very interested in talking about these ideas. I will repeat some and talk a little bit about a new review. And this is work that builds on a review together with collaborators Erika Larsson and Emily Moore, as well as some work together with a former postdoc. Homo papaliasti mainly. So since we're in the gene expression session today, I want to start out by saying that hybridization is common, and that the revolution of sequencing has basically shown that it's an important way, or an important evolutionary force contributing novel variation for selection to act on. And hybridization may generate variation in a way that's different from mutation. So for instance, hybridization can transfer large co-adapted gene complexes that have already been tested by selection in a similar genomic background. And hybridization has also been shown to enable different types of evolutionary responses, such as heterosis and transgression, that I will come back to. And hybridization can create novel variation in a bunch of different ways. So for instance, there can be novel combinations of different parental alleles that lead to mosaic new phenotypes, such as in the heliconius butterflies here, for instance. And hybridization can also come up with phenotypes that are more extreme in some traits than both parental species, transgressive, if there is complementary action of additive genes, basically, such as in the sunflower example here, where the two parental species have plus alleles for tall growth for different genes, and that this can then lead to an overall higher number of plus alleles in F2 and later generations. But today I'm going to talk about some ideas of whether altered gene expression or altered regulation of gene expression could lead to extreme patterns of gene expression in hybrids. And I would very much welcome any discussion of this. So gene expression has long been known to be very funky in F1s. And Emily Moore, who has a poster tonight, poster board N18, please see that poster. It's going to be lovely figures that she's made. And Erika Larsen and I, we reviewed the evidence for gene expression in F1s and other early generation hybrids. And we found that in 40 out of 52 studies of sterile hybrids, so a high majority, there was actually transgressive gene expression. But interestingly, in more than half of the fertile F1s, we also found extreme patterns of gene expression. And even more intriguing, I think, is that we actually find evidence of transgressive gene expression also in stabilized hybrid lineages or species. So there's 3 flower examples, then there's a sparrow. Sorry, no image for the Drosophila in this version, but it looks like Drosophila. And there's also evidence in a 200 generation of hybrid fish. And here we cannot assume, as in F1s, that this gene expression is misexpression that could have severe fitness consequences. So I think this is very interesting. And I'll start with going through one of the case studies a little bit. So the Italian sparrow is a homoploid hybrid species. And Glenn Petersetre and his lab have basically been showing that it originates from cross between the house sparrow, which you surely all have seen tons of all over Montreal here, being invasive, and the Mediterranean Spanish sparrow. And this initial hybridization event has taken place more than 5,000 years ago. So there's been ample of room for recombination and selection to form a functional hybrid genome. And seeing the number of house sparrows here in Montreal and elsewhere in the world, those Italian sparrows had to be pretty fit in order to actually outcompete this parental species, I think. So my former postdoc Homo papaliasti and I, we went, and some other collaborators, we were looking at gene expression in the testes of these Italian sparrows, and we compared it to the parent species. And since the Italian sparrow has an intermediate genome, maybe we could expect that the gene expression would not be more extreme than the parent species along any of the axes, and that's here illustrated in yellow. But what we found was instead that, let's see if we have a pointer here. Well, no. So what we found was instead that we had a high hybrid Italian sparrow to parental species ratio both along the x-axis and the y-axis for a large number of genes. And we also found genes that were lower in, or were expressed in lower numbers in both the Italian sparrow in the hybrid and in both parental species. And if we look at the genes that are overexpressed in the Italian sparrow compared to both parental species, it basically makes sense in terms of what we know of the Italian sparrow. So for instance, we find a high number of genes that are involved in the mitochondrial respiratory chain, cytosolic and ribosomal mitochondrial proteins. And this is potentially a response to the mitonuclear incompatibilities that we have been recovering in the Italian sparrow. But this is nothing we've gone further and tested. And it's also interesting to see that there's a lot of those genes that are involved in binding of the sperm to the zona pellucida of the egg that are being more expressed in the Italian sparrow than in both parental species. And those genes have been shown to be under selection in the parental sperm proteomes in a separate study. And I'm going to show you a different representation of this data here. So in the left hand of this figure, you can see that in the testes, there's approximately the same number of genes that are differentially expressed compared to the house sparrow on top and to the Spanish sparrow on the bottom. However, if we look at the ovary instead, there's almost no genes that are differentially expressed compared to the house sparrow, but a lot of genes that are differentially expressed compared to the Spanish sparrow. So I think this pilot study just leaves us with a lot more questions than answers, and I think that this entire background of observation of strong misexpression or strong transgressive expression in F1s and early generation hybrids, but also stabilized hybrids, begs a lot of interesting questions. And some of the consequential ones would be what regulatory mechanisms can lead to altered hybrid gene expression, because in F1s we have an intermediate composition of regulatory elements, and we don't expect that it's going to be like an overall higher number of like plus alleles as in the sunflower example before, already in the F1 generation. And we don't know whether gene expression changes at a gene-to-gene or a network level, or if we can use parental patterns of expression to predict hybrid gene expression of specific genes. And I think an exciting one is whether hybrid gene expression could indeed be, maybe some extreme patterns of expression could be adaptive in novel environments, for instance. Or if it's just basically the hybrids that are trying to regain fertility through upping some of the genes that they are expressing. So in my current work, or in the work that my lab is doing currently, we are using 3 different Italian sparrow populations on different Mediterranean islands to try to gain more general insights to how gene expression evolves in hybrids. And we are using these populations because we have been running whole genome analysis on them. And here is a PCA representing whole genome data. And as you can see, there's a house sparrow, one of the parent species in blue on the left hand, and the Spanish sparrow in red. And you can see that those populations basically have different proportions of these 2 parental species genomes. And here is a clustering analysis basically making the same point. So some of the populations have a higher probability of belonging to the red Spanish sparrow cluster and some to the blue house sparrow cluster. And if we look across the genome, we cut up the genome in small bits and pieces, and we produce one phylogeny for each one of these pieces. You can see that it varies very widely across the genome. So this gray blur here each corresponds to like one part of the genome. And the darker black lines, they are basically just the most common trees, not like a true tree. So based on this genomic background, we think that these are going to have some independent component to their genome stabilization. So in the current projects, we are using these to look at gene expression in all of those populations. And I would very much encourage you to look at Alex Lawrence's poster. It's going to be on the 29th, board G16. So we have been sampling 8 tissues, I think, approximately 8 to 12 individuals per each one of these Italian sparrow populations and the parent species. And we've been sampling them in a common garden at Lund University the day after the first bird is laid, which is more challenging than we would expect from the start. And we are going to use this data to address questions like, can we predict whether which genes are going to be transgressively expressed, or how genes are going to be expressed in the hybrid based on the network the gene belongs to, the function the gene has, whether the gene is mainly cis-regulated, mainly trans-regulated, or cis-trans compensatorily regulated. Is there a role for mitochondria, sex chromosomes, germline restricted chromosomes potentially? And then, of course, there's multiple layers of regulation of gene expression, and there's many phenomena in hybrids which include, for instance, structural changes such as inversions, insertions, deletions. There's new releases of transposable elements with new insertions and new expression. We know that epigenetic regulation, both histone modifications and methylation, can change in hybrids. Postdocs Kalle Tunström and Rachel Stewart in the lab are currently working on these questions, assembling new reference genomes and so forth. And they will both have talks today during the conference, but on other topics, so adaptation 9 and genomics 14. And I want to leave you guys with what I think is an outstanding question, and this is basically, can gene expression provide an additional mechanism for like novel variation that selection could act on to give rise to adaptation from hybridization? And I just think it's so intriguing how 17% of the F2s resulting from 7 different types of yeast crosses in different extreme environments were actually more fit than both parental species. And of course there could be heterosis in this, and this hasn't been coupled to gene expression. But that we know that hybrids can be more fit in novel environments, and we know that they also have funky gene expression, I think basically begs us to try to do the hard work and look at gene expression. And maybe we should start with something that's not as complicated as sparrows. Yeah, and I would be super happy to take any questions and to have feedback and discussion. And I want to thank my co-authors and the amazing team that has been doing all the hard work with getting those sparrows and getting them sampled. The sparrow group in Oslo, where I did my postdoc, and some funding. Thank you. Okay, so we have 1 minute, maybe one question.
Expression differences in testes are more pronounced than in other tissues.
So we only have testes and ovary, but one of the things that I'm thinking is that the W chromosome in females is probably inherited from the same parent as the mitochondrion, and that could be an important. And they have the house sparrow mitochondria, and it's also the house sparrow that has a more similar gene expression. But I really think we need to do better sampling. So basically, I have a freezer full of this better sampling now. So I'm hoping to be able to give a better answer in the future.
Hello, can you hear me okay? So today I'm going to be talking about a project that a graduate student in my lab, a PhD student, Yihan Wu, has been working on, called Genome-Wide Identification and Analyses of Copy Number Variants, which I'll call CNVs for short, and 751 populous trichocarp individuals from natural populations. So what are copy number variants? Copy number variants are a type of genome structural variant where individuals show variation in the number of copies of a small segment of the genome. And that segment actually is more, it could be more than just a small segment, it could be of varying size, and indeed some of the ones that I'll show are actually quite large. Typically people define these as greater than 50 base pairs, but sometimes up to several kilobases. People have been interested in these in a lot of different study systems, and they've been studied in different kinds of contexts, especially in agricultural plants, looking at different lines of agricultural plants. They've also been studied in outside the plants and animal systems too. And also, people are quite interested in how these copy number variants affect gene expression. So that's kind of where this talk ties into this particular session. So the copy number variants, these can form through a number of different ways. Some of them are homology-mediated mechanisms, which means that they require some sequence similarity in order to occur. So these include non-allelic homologous recombination, which is shown in the figure over here. It's also sometimes called unequal crossing over. They can also form by microhomology-mediated end joining, and also by a single-stranded annealing. A lot of these mechanisms were worked out in mammalian systems, but they also seem to be taking place in plants too. There can also be non-homologous end joining as another type of mechanism that's not a homology-mediated mechanism. Also through replication error and slippage, especially in the high repeat regions such as tandem repeats. And in addition, they can also be mediated by transposable elements. Now I'd like to introduce the study system that we used in this study. This is Populus trichocarpa. It is the cottonwood tree. If you live in places, especially in North America, where we have a lot of cottonwood trees, typically during May, if you see these little seeds, they're very tiny seeds that have little cotton-like fibers, and they're wind dispersed. Like, for example, if you ever walked across a sidewalk under a cottonwood tree, or if you parked your car under a cottonwood tree, you probably have seen these. The Populus trichocarpa, these are used a lot in paper production, especially hybrids that grow quite rapidly. In British Columbia, where I live, they are very important from the perspective of growing them for paper products. There is a reference genome that was first published back in 2006. So it was one of the first few plant genomes that was sequenced. And these are native to British Columbia and Northwest Washington, this particular species. There's also another species, Populus balsamifera, that's spread across a wide range of North America, but across Canada and parts of the United States. There's been previous studies of local adaptation in Populus trichocarpa. For example, there's been shown to be, from other studies, a latitudinal decline in physiology as well as phenotypes. There's been a lot of this work going on at UBC, University of British Columbia, where I am. There was a project called the PopCan project a few years ago that was funded by Genome Canada, where they generated a lot of whole genome sequences from populations across the range, and that's one of the major data sources that we used in this particular project. Also, various studies have shown that multiple single nucleotide polymorphism, or SNP candidates, are linked to adaptive phenotypic traits, as well as physiological traits and also developmental genes. So the genotypes that we used, these are genotypes that were used by the PopCan project, as well as some genotypes that were used in a study by the Joint Genome Institute in Tennessee. We used a total of 751 genomes, and when I say we, I actually mean Ihan Wu, my PhD student, because this was her project. I also like to note that Ihan was the one who conceptualized the project, and she's been a really major driving force of the project, so I'd like to give her credit for that. And you can see that these populations are spread across the range in British Columbia, with an emphasis on ones that are close to close to the coast or within the mountain range, as well as down into Washington State. So some of the questions that Ihan asked in the project was how much and what are the characteristics of copy number variation and other genome structural variation that exists within Populus trichocarpa. No one had identified this on a population-wide scale. There had been smaller studies of copy number variants in like a very small number of individuals, but nothing on the scale of what Ihan had done. In fact, there's actually in natural populations, there aren't any studies in plants at this particular scale. Now, in agricultural plants, people have looked at this on a large scale in different cultivars of certain agricultural plants, like rice, for example. In terms of other questions, we're interested in getting insights in terms of mechanisms that led to the formation of these copy number variants based on analogous studies in humans. We were also interested in asking which processes are enriched in genes within the C and B containing regions. So what genes do you more commonly find in these C and B regions? And finally, and connected to this symposium, is what effects do the copy number variants have on gene expression? So I'll show just a little bit of data from the project. I don't really have time to show all the data, but I'll just show a few selected figures and tables from Ihan's project. First of all, this is the number of variant types. So Ihan, we were particularly interested in the duplications, and oops, sorry about that. You can see that there were close to 17,000 duplications that were documented. There were more deletions that Ihan found. There were also a smaller number of inversions and a fairly large number of mobile element insertions. So in terms of sizes, this is a graph showing the size. As you can see that the vast majority of them were relatively small. Most of them tend to be about 500 bases or smaller, but there were a lot that were, or quite a number that were quite a bit larger. This graph stops at 2,000 bases. There were a few that were larger than that, but it's not shown on the scale just so that you can see the distribution of the others better. Here's a distribution across the different chromosomes. There are 19 chromosomes in Populus trichocarpa, and one of those chromosomes also has a sex locus. And here you can see the coverage across the chromosome. It's fairly, I mean, there is some variation, but it is fairly consistent. And as well as the numbers, there's also some variation across the numbers, but nothing really major differences, except the chromosome 1, curiously, does have more. In terms of copy number variant links, this is a graph showing the different types of copy number of different structural variants that we studied, including deletions, duplications, inversions, and mobile element insertion, which is TEs, is the MEI. And one thing that you'll notice is that generally there are more deletions, as I showed on one of the previous slides, but also you'll notice that in the larger size class, for example, this 1,001 to 5,000, the number of duplications relative to deletions is actually increased than it is in the smaller size class, which was kind of interesting. This is a Go term analysis that Ihan did to look at Go term enrichment. Some of the ones that were enriched were protein localization, amino acid activation, RNA amino acetylation, tRNA amino acetylation, regulation of organelle organization, etc. So these show some of the Go terms which are enriched in these C and Vs. Now, of course, I do realize Go terms are fairly general, and they give sort of a general idea of function, but they don't tell you exact function. So obviously, further study needs to be done of interesting genes in that regard. Ihan also had a very interesting idea that she got from reading the human literature about inferring formation mechanisms, and this was to compare the occurrence of C and Vs against regions of low complexity, and to compare against TEs, as well as comparative sequence similarity around the copy number variants. And what she found was, here's a summary of the results. So the largest number of these seem to form by non-homologous recombination, and the evidence for that is through high similarity in the sequences neighboring the start and end of the C and V. Now, this doesn't prove any of these. We haven't proven the mechanisms, but we are getting some clues as to what they might be. The next highest category seems to be a formation by variable number of tandem repeats. The next, which was relatively small compared to the others, was non-allelic homologous recombination. Okay, now the expression part. We were definitely interested in looking at expression variation due to C and Vs, and my lab has studied expression a lot in other contexts, especially in the context of polyploidy plants. So looked at transcriptome sequences from 423 genotypes in leaf and xylem that were generated largely by the PopCan project previously at the UBC. And it was nice because there were two different tissue and organ types, the leaf and the xylem. She did requantification with salmon and examined only the cis effects using genes that directly overlap with the C and Vs. So here's an example of results. She was interested in looking at whether or not there's been negative or positive correlation with expression. Our hypothesis was that there would be mostly a positive correlation of expression, but we didn't really find that to the extent that we thought we would. In terms of duplications, yes, there were a lot more that had a positive influence or positive impact on expression, but there were also a fairly substantial number that had a negative impact on expression, which was not what we expected. The deletions were fairly equal in contrast. In terms of the go terms, this is an example from leaves. We have a response to various types of salt stress, osmotic stress, cadmium ions, inorganic substances. Basically, response to abiotic stresses seem to be quite common in terms of the overrepresentation of go terms in the leaf. In the xylem, it was really different, though. Some of the top go terms were coenzyme biosynthetic process, nucleoside triphosphate metabolic process. Basically various metabolic processes seem to be the most common in the xylem, so quite different from the leaf in terms of enriched go terms. And some of these genes would be quite interesting to look at in more detail in further studies. Okay, so finally I'd like to acknowledge Ihan Wu, my PhD student. I'd also like to acknowledge Professor Quentin Cronk, who has been a very valuable member of Ihan's thesis committee and also for valuable discussions. The data come from the PopGen project at UBC and also the Joint Genome Institute at Oak Ridge, and we got those from NCBI, also my funding source and computational source. And finally, this is a picture of Ihan. Thank you.
All right, it's now officially 3:30. So good afternoon, everyone, and thank you for coming to my talk today. My name is Austin Biddy, and I'm a PhD student at the University of Alabama in Birmingham. And the title of my talk today is Differential Gene Expression is Constrained by Clade and Ecological Divergence in a Baja California Endemic Lizard. So a process of interest for this study is population isolation, divergence in microclimates, and principally this can happen whenever gene flow is impeded by spatiotemporal processes such as physical barriers between populations as well as seasonality of rainfall, for example, or differences in weather or biological events where this mismatch can result in individuals not interacting with one another and thus impeding gene flow between them. So if these populations are isolated, then we should see adjustment to dissimilar microclimates via gene expression data, and especially novel pressures and or acclimation to these different environments, and the expectation is to see differences in gene expression overall between the different populations. And these changes can have ramifications for behavior, phenology, and gene flow, feeding back into isolating these populations over space and time. A good area to study spatiotemporal isolation is and how this relates to expression differences of the Baja California Peninsula. There's been shown to have interspecific divergence across a variety of taxa along this north-south axis. And there's heterogeneity of microclimate and topographic heterogeneity. So this PCA plot was created using occurrence data from sampled locations, 19 bioclimatic variables, and solar radiation. And you can see between the north and the south populations, there's divergence in their microclimates. And even within the south, we can see divergence across populations. And I'll show a map later that might make a little more sense in terms of this spatially. So there is microclimate divergence across the peninsula. And while there's plenty of population genetic studies about the Baja California Peninsula, especially with mitochondrial data, differential expression studies are lacking, and this can provide compelling insight into Baja California endemics and how this relates back to this diverged microclimate that we see across the peninsula. And a species that kind of fits this mold is the Urosaurus hygrocontis, which is the black-tailed brush lizard. It's endemic to the region and has deep mitochondrial divergence, forming two north-south clades, principally. Biologically, they're arboreal and insectivorous, which in an otherwise arid landscape, and they have diverged climatic niches between the north and the south, and even more so 3 climatic niches in the south by itself. So there's a lot of climatic heterogeneity. So the hypothesis is that these two clades show differential expression and processes related to overall ecological differentiation. Sampling took place, of course, along the Baja California Peninsula. 31 individuals were sampled. We sampled the tails because it's relatively non-invasive because they can be regenerated. And we sampled 15 individuals from the north and then 16 from the south across 2 populations in the north and 3 populations in the south. We used RNA-seq to create a transcriptome of these mRNAs. And fortunately, somebody in our group annotated the genome and published it. You should check it out. And that makes transcriptomics rather easy to do. And then with these transcripts, we perform Samuel quantification of the reads, DE-seq2 for differential expression, and then GoTerm enrichment to look at what these genes from a broad perspective do. And for the statistical design of the differential expression, we controlled for sex, since that's pretty common in reptiles, and that way we can only look at variance between the northern and southern populations. So of these differentially expressed genes, there were a total of 671 between the north and the south relative to each other. When looking at genes with a log fold change above 2, there were 71 between the North and the South. And in total, there were 363 go terms that were these genes were associated with. And then I have a Revigo tree map off to the side here, and you can see that and this is just for biological processes, not cellular component or molecular function. And you can see that there are plenty of genes that are clustered together. And I pulled out some of these larger terms to get them out of the way and show you what these other genes do. But because there's 363 go terms, it kind of makes it difficult to disseminate each one and understand how this relates back to the ecology of this lizard. So we just looked at the go terms of interest. So the 3 that we chose were mitochondrion, lipid metabolic process in response to abiotic stimulus, with the hope that this can help us understand this ecological differentiation of these lizards. And I do want to point out that some of these are rather general, like mitochondrion, and to make sure that this represented all the genes that could be associated within our data set. With the mitochondria, we compared things like mitochondrial membrane and cellular respiration, et cetera, and to make sure that this go term encompassed all of those genes that kind of fell under that umbrella of what the mitochondria does, and also had unique genes within itself. So I have a PCA plot right here. So you can see there are patterns of clade divergence of gene expression between the North and the South populations, which was to be expected. However, there was some overlap, but it's still interesting to see that these wild populations do show natural variation in gene expression between them. So also, in addition to the PCA, the heat map is clustered by clade. The rows show genes, and the individuals are columns. You can see there's clear clustering of North and South along this first grouping. But as you kind of go further down, and I want to point out that the orange is upregulated relative to one population. The indigo color is downregulated relative to the other population. And you can see that there's clustering by North and South, but if you look down toward the bottom, that these 2 southern populations that are separated by this black line kind of show some dissimilarity between each other. And this could potentially be because of, the animation went backwards. So here's the box kind of outlining what I'm mentioning. And some of these genes are also close to 0, which means they're negligibly differentially expressed. But once again, they show microclimatic divergence and differences, so it can be kind of expected that some of these individuals might not reflect the entire southern clade because they have different climatic niches from where these individuals were sampled. So to kind of pick through these 3 go terms that I have shown to you in a previous slide, populations do differentially respond to abiotic stimuli. I have the go ID here just for reference for yourself, but response to abiotic stimulus is basically just response referring to how variations in temperature and precipitation facilitates changes in gene expression. So first I want to discuss the northern population. So some of the main functions that were pulled out when we looked through all the genes and tried to categorize them were angiogenesis and hypoxia. So angiogenesis is the formation of new blood vessels, and hypoxia is low oxygen. And these can be sometimes associated with oxidative stress, which is a result of mitochondrial efficiency, or can be a result of mitochondrial efficiency. And they also had a lot of gene functions that are associated with vasodilation and vasoconstriction, which are associated with metabolic demand, or can be associated with metabolic demand, whereby vasodilation occurs, so more blood can flow and deliver more oxygen to tissues and supply more oxygen whenever metabolic needs are present, or they can be constricted, doing the inverse where they're trying to reduce blood flow and reduce the amount of oxygen because it's not necessarily needed at that tissue type. And so both of these have to do with metabolic demands, which is a pretty interesting theme we'll cover for the next few slides. And then in case any of you are gene people, here are some of the genes that came out from DE-seq, mapping back to the annotated genome. So we have ANGPT4, which is involved in endothelial cell survival, migration, angiogenesis, VEG-FC for growth factor, which is active in angiogenesis, and then ED-NRNA, which has a role in vasoconstriction. So trying to show you some of the genes that have these associations that are being discussed to make a more tangible result for you. Now let's move on to the south. So the southern Baja Peninsula is hotter. This mouse is kind of sensitive. Okay, my laser pointer went away. Okay, that's okay. I've timed myself so I can make those mistakes and know that I'll hit the 12 minutes. So the Baja California Peninsula is hotter, and that could potentially lead to individuals being more exposed to UV radiation. Thus, it's unsurprising that we have DNA damage repair showing up as a common gene being associated with this region. Furthermore, there were a lot more heat shock proteins and chaperones or protein folding genes that were upregulated in the south relative to the north, which could also potentially be involved with heat stress, as well as the chaperones and protein folding to make sure that these proteins that are being translated aren't resulting in cellular dysfunction. And then once again, if you're gene people, one of the ones that were differentially expressed was heat shock protein binding factor, HSPB1, which is heat shock protein. It functions as a molecular chaperone. DNA, JA4 for chaperone binding and unfolded protein binding, and then CRIAB, which is also chaperone-like activity under stress conditions. So now moving on to lipid metabolism, which is one of the Go terms I had shown earlier. So one lipid metabolism, or so lipid use varies by clade, and one of the lipid metabolism genes or functions that kind of poked out being kind of common across both populations was fatty acid oxidation. So fatty acid oxidation can occur whenever there's physiological stress or dietary constraints, and these organisms need to kind of weather some starvation, periods of starvation. And so when there's paucity of food, they'll break down these fatty acids, use them for energy supplement, and this can also have ramifications for things like reproduction or dispersal, so it can overall be potentially linked to population ecology and just for them trying to avoid starvation. But I want to kind of go to the inverse of that, where in the south we're seeing increased precipitation relative to the north, and since these are insectivorous, then potentially if there's more precipitation, we'll see more foliage, more habitat for insects, and thus there's more prey items for these lizards to consume. And so unsurprisingly, we saw more lipid biosynthesis genes that were upregulated in the south relative to the north, and that could be involved with this nutrient abundance and thus their ability to, or their fortunate ability to synthesize more lipids to use later on in periods of starvation that might occur. And let's see. And then I also do want to point out before I show you these wonderful genes again, is that the tail was the tissue that was used, or several tissues that were used, and this is the lipid storage depot of the lizard. So it's very important for storing lipids as well as using those whenever they're needed for times of starvation or insulation. So CYP2J6 is involved in unsaturated fatty acid oxidation, DGKA for biosynthesis of complex lipids, and then GPAT4, which is involved in lipid biosynthesis. And once again, if you're a gene person later on, you want to talk about them, if you know them more intimately, I'd be more than happy to discuss these genes with you. And then lastly, cellular respiration is more differentially expressed in the south. So if these respiration genes are increased in the south, it leads to the question, of, is there more ATP that's being generated? And if there is more ATP, how's that energy being allocated amongst the populations and between the populations? And how can this potentially affect a life history repertoire, such as body size, growth rates, age of maturity, lifespan, and reproduction? And also, if there's a mismatch in energy allocation between these two populations, does that have potential ramifications for these differences in life history traits, how that affects their overall population ecology, metapopulation dynamics, and is this a potential reasoning for genetic divergence that's seen in the mitochondrial data of these species along the Baja California Peninsula? And then last few genes I want to talk about, which MTATP6 is involved in mitochondrial ATP synthesis, COX6B1 for electron transfer, and then FMC1, which actually is kind of like a jack-of-all-trades gene of the mitochondria, but it is also involved in the assembly and stability of the mitochondrial membrane ATP synthase. So some overall takeaways. There is divergence in microclimate, not just between the north and the south of the Baja California Peninsula for this lizard, but also within the southern populations, which is also reflected in some of the gene expression patterns that we see. In response to abiotic stimulus, we're seeing differences potentially based on temperature, such as DNA, angiogenesis and hypoxia, as well as DNA repair and from UV radiation potentially, as well as heat shock proteins. And then from the south, thinking about fatty acid oxidation, dietary or prey items being available in lipid biosynthesis. And then lastly, once again, tying together how metabolism might result in this population divergence that's seen in the mitochondrial data. So with that, I will pull up my acknowledgments for the Baja Geogenomics Consortium and the wonderful people that are involved with that, the Dolby Lab, and then NSF for funding this project, as well as my ability to be here today. And with that, I'll take questions.